Now, topic 3 is the antigen-antibody interaction. This is something which most of us have experienced. If you happen to go and check your blood group, you can see that the process for the identification of blood group is just antigen-antibody reaction. The process for the identification of malaria parasite using strip is antigen-antibody reaction. The process for the identification of either if you are HIV positive or negative is just antigen-antibody reaction. So we are going to understand the basic principle behind that. Now, we already explained that the antigen has an epitope, which is a binding site or receptor site, whereas also the antibody has a binding site, which is called paratope. So collectively, they can join together. Now, for us to understand the basic principle behind the antigen-antibody reaction, you have to know that there is a situation where there is neutralization, and there is a situation where there is kinetics of the antigen and antibody. How strong are they binded together? How strong can they dissociate? So there is issue of how strong they can associate and how strong they can dissociate. And these can be determined by their valency or their kinetics. If I say valency, is how many antibodies are there that can bind with the antibody, or vice versa. So that's why the antigen-antibody reaction is just a biochemical reaction between antigen and antibody. Specific antigens, they can bind to specific antibody when they come closer to a distance in a several nanometers. There is properties or situation that can determine how much this antigen and antibody they can bind together. One of them is highly specific reaction. In a situation where the antigen and the antibody, they all have the compatible receptor site. So how specific and how strong are they going to bind together also determines that. So that's why we have highly specific reaction. So all the reactions that has to do with antigen antibody, it must be specific. If there is reaction, then there is specificity. Number 2 is that all the antigen antibody reaction, they behave in an observable manner. That's why you can see agglutination when you mix your titer values with a blood to identify blood group. You can see the agglutination. When you want to identify whether there is typhoid fever or not, you see agglutination. When you want to identify whether there is HIV or not, you see strip line, formation of the strip line. That strip line is indicating that there is antigen antibody reaction via agglutination. So they occur in an observable manner. Number 2 is that the reaction is non-covalent interaction. Non-covalent in the situation where it is reversible. There are certain reactions that they are covalent, they are entirely not reversible. But in the case of antigen antibody, they can dissociate, therefore they are non-covalent interaction. Number 4 is that there is no denaturation of antigen and antibody. In some certain chemical reaction where there is reaction between two substrate, there is entirely denaturation of the substances. Sometimes there is thermal reaction, production of energy and heat. That means there is denaturation. But in the case of antigen antibody, there is no denaturation. And number 6, it is reversible. That's why I say that they can be dissociated by some certain situation or factors. So it is a reversible reaction. Then how are we going to determine the strength of the antigen antibody reaction? First of all, we have what is called the affinity. Affinity is the strength of binding, forward binding between one antigen and one antibody. So when one antigen and one antibody come together, affinity tells the strength at which they are binding together because of their specificity. Then number 2, there is avidity. Avidity is testing the strength of binding between many antibodies with many antibodies. So the overall strength of the antigen-antibody complex, it is called avidity. It depends on the affinity valency and the structural arrangement of the epitopes and paratope. So affinity valency means how many antibodies are binding with how many antigens. There is a situation where there is a lot of antigens but smaller number of antibody, or vice versa, a situation where there is high number of antibodies but smaller number of antigen. So that is termed the valency of avidity or the valency of affinity. Then there's cross-reactivity. This refers to the ability of an antibody to bind to a similar epitope of different antigen. So it is called a cross-reactivity, the ability of an antibody to bind to similar types, epitopes of different antigen. So we have, let's say we have two antigen, they have the same binding site, but they are different antigen. So the antibody can be binded together to two category of antigens. So it is called cross-reactivity. Then down to the kinetics of antigen-antibody reaction. How rapid initial combination, how rapid is the reaction during the initial combination? So when they come together to bind, how rapid are they going to bind together between the antigen and the antibody? It can be short and it can be quick. It depends on the prevailing factors, either temperature, either other factors that they can affect the rate at which they can bind. And another thing is that it is reversible. The antigen-antibody association can be dissociated into antigen-antibody based on the prevailing factors. And number three is the dependency on the concentration. The higher, that is the valency. How many concentrated antibodies are there to bind to the how many antigens available? So this can be defined by the valency and the binding site on both the antigen and the antibody. Now the law of mass action. The law of mass action is saying that the rate of chemical reaction is directly proportional to the concentration of the reactant. So the rate at which the antigen antibody bind depend on the concentration of the antigen and the concentration of the antibody. Where the concentration of one of the two is low, the rate of the binding will be slow. Where the concentration is equivalent or at equilibrium, then the rate of the concentration will be extremely strong. So vice versa, that's why the law of mass action is also applicable in the antigen-antibody reaction. The rate of chemical reaction is directly proportional to the concentration of the reactant. Now, the factors affecting this antigen-antibody reaction. Temperature matters, because wherever there is temperature, there is tendency for denaturation. So at some certain temperature, it can aid in the strong binding between the antigen and antibody. So that's why temperature, wherever there is a binding between matter or substances, temperature play a very vital role. Number two is pH, hydrogen concentration. At optimum hydrogen concentration, usually 0.5 pH to 8.5 pH, which is around neutrality to alkalinic temperature, pH, there will be optimal reaction between the antigen and antibody. But at alkalinic or halophilic pH, it may slow the rate of the reaction between the antigen and the antibody, and vice versa between the alkalinic and the acidic pH. Now in number three, we have ionic strengths. This can be applicable to sodium or chloride. That's the saline solution. So the higher the sodium, the lower the antigen-antibody reaction, and the lower the sodium concentration, the higher the antigen-antibody reaction. And there is enzymatic treatment. A lot of proteolytic enzymes used to enhance the antigen-antibody reaction. Proteolytic enzymes are highly proteolytic in nature, and they can aid in the increasing the rate at which the antigen and antibody combine together. Number four, we have the concentration of the antigen and antibody, which we have already explained. That is the affinity kinetics and the avidity kinetics. Whichever the concentration of each of them can aid in how strength the reaction can take place. Then there is the binding site. You can recall about the heterophilic reaction. A particular antibody may bind to two antigens because they have the same binding site. So that's why the number of antigen binding site also matters. Then there's also the structural arrangement, where depending on the types of antibody, if it is IgE or IgD or IgE, they are all monomers and they have one binding site. But in a situation where it is IgM, it is a pentamer, and therefore the binding site and the structural arrangement can matters and can play a very vital role in the antigen-antibody reaction. Now, stages of antigen-antibody reaction. Stages of antigen-antibody reaction can be either rapid, reversible, or without any visible effect. That means it is extremely negligible to the extent that you cannot even notice the reaction. So at the primary stage, if the valency is the same, it will be rapid. And by action of either temperature or other factors like pH and the valency, it can also cause the reaction to be reversible. Then where the valency is not the same, then the visibility of the reaction can be entirely negligible and not visible. Then second stage. Second stage, this is usually applicable to B cell before the production of the memory cells within the body, that is in vivo. In vivo, we can have a lot of reactions like agglutinations, the clumping between the antigen and antibody. There is precipitation, that is individual affinity reaction between one antigen and antibody within one solution, within the blood or within one solution if it is outside the human body. Then there is complement fixation. A complement formed by complement system to destroy a particular antigen. That is complement fixation. Then there is neutralization, where the concentration of the antibody supersedes the concentration of the antigen. So it's like mixing a concentrated substances with a water. You are going to neutralize it. So that is neutralization reaction. Then there is immobilization, a situation where a conglomerate concentration of immune cells, they can immobilize a particular antigen in either a tissue or in an organ. This is usually happening in the case of parasitic infection or in pus formation. Then there is opsonization. Opsonization is just a situation where a particular immune cell presented an antigen. If you take example of CD4 or major histocompatibility complex, they are used in opsonization. They seemingly binded with the antigen and presented the epitope of the antigen on their surface so that B cells can recognize it, then they can trigger the production of memory cells using plasma B cell. So thank you for listening, and later we are going to continue with the more response on that topic 4.